All right, so this lesson is a continuation of the male reproductive system talk that we covered, or you covered with Professor Fink in your last lesson together. We are going to finish up talking about the male reproductive system in terms of spermatogenesis, so actually making of the sperm. In the last lesson, you learned a little bit about what cells secrete testosterone and make sperm and everything. And so that was kind of an overarching picture of the male reproductive system and its structures. But today, let us start talking about how we actually make those gametes. And so a gamete is a haploid cell, so one copy of DNA as opposed to two. In males, this is sperm. In females, this is eggs. And so we will talk about this process in males and then go right into talking about this process in females. Hopefully we'll get through all of that today. And so as we see at the top of the screen here, the process of spermatogenesis is literally generating sperm. Okay, and so this process involves meiosis. And so I posted a couple videos to remind you what meiosis is. It's not critical for you to be able to tell me where all of the different chromatids are going and when crossing over and all that kind of different stuff happens. But you should have a general idea of what meiosis looks like and how it's producing individual unique haploid cells. That is haploid one copy of DNA, and they're unique in that each sperm has a different combination of genes. Okay, and so the process of meiosis is responsible for making millions of unique sperm a day. Okay, so this process begins with something called a spermatogonium. Okay, and spermatogonium is the singular form, right? So it is a stem cell. It is diploid, so two copies of DNA. These two copies of DNA ultimately have come from the parents of this individual. Okay, so we're kind of talking about multiple generations here. So as we walk through this process, I'm going to be talking about spermatogenesis happening in Papa, right? So this one individual, his genes ultimately came from his parents. So grandpa and grandma, their genes combined to make his DNA, his genome. And so their, right, grandma and grandpa's DNA combined to make the genome of the Spermatogonium. And so that is unaltered in theory, save for any mutations or whatnot, in Papa, right? So in the person that we are talking about now, that is making sperm. Okay, these stem cells, just like any other stem cells, have to undergo mitosis. Remember that mitosis is different than meiosis. Mitosis is the process of copying a diploid cell to make an exact copy, or actually two copies. Okay, so mitosis is just making more of these stem cells. And just like we've seen many times in the past, right? So for example, in the stratum basal of the skin, we have those stem cells making more and more of themselves, and all of the daughter cells are going to be clones. Okay, and so of the two daughter cells, okay, one of them is going to stay behind and it's going to remain a spermatogonium. Okay, so we have to replace these stem cells, and at the same time, we are making more of what are called type B cells that are going to complete the process of meiosis. Okay, so this here is a summary of meiosis. Okay, up here, this is representing a spermatogonium. Right, a germ cell, that is, it is the stem cell. It is responsible for making all of the sperm ultimately. Okay, so these guys up here are going to divide, right, making one cell that remains a spermatogonium. The type B cell is going to proceed through meiosis 1 and then meiosis 2. And ultimately, these two processes together are going to produce 4 unique gametes. Right, so haploid cells, one copy of DNA, because ultimately these cells are designed to fuse together with another haploid cell, the egg, to create a new genetically unique offspring individual. Okay, so just a couple key points about these processes. Again, I am not going to ask you to follow individual chromatids and talk about cytokinesis and spindle fibers and everything. None of that is critical for this part of the class. But you should be aware that in the beginning of meiosis 1, right, in the step called prophase 1, a process called synapsis, or crossing over, is going to occur. And essentially what's happening here is that the chromosomes from grandpa and the chromosomes from grandma are going to line up, right? So they're going to replicate, they're going to line up, and ultimately they are going to wrap around each other kind of like a Twizzler. And some of those genes, right, or in some cases, like half of the chromosome is going to literally jump over to another chromatid, right? So this is actually mixing up grandma and grandpa's genes so that baby, right, the offspring of this individual is going to be unique. It's not going to look exactly like the papa. It's not going to look exactly like grandma or grandpa. It's going to be a completely unique individual. Okay, and so one of the reasons for that is that in the very beginning of meiosis 1, we mix up the genes. Okay, so here is the gene for lactase from grandma. Here is the gene for lactase from grandpa. We are going to switch them on individual chromatids. And so again, that's called synapsis, or crossing over, in the very beginning. There are a couple other places within meiosis that are going to ensure that we get 4 unique gametes, right? 4 unique sperm. The next place is in anaphase 1, and this is called independent assortment. So essentially, different chromosomes are going to line up in different parts of what is called the metaphase plate, and just completely by chance, they're going to line up, and these ones are going to be pulled in this direction, these ones are going to be pulled in this direction. So ultimately, both of these daughter cells are going to become different sperm. Okay, and so again, it's completely random where these things actually line up, and where they line up is going to determine which daughter cell, which sperm they're going to be part of. Okay. All right, this process happens kind of again in anaphase 2. Again, we get this independent assortment along the metaphase plate. And so when these final chromatids are pulled to opposite sides of the cells and ultimately into their own genetically unique cell, each genome, each haploid genome or copy of DNA is going to contain a different combination of grandma's genes and grandpa's genes for baby. Okay, and so take-home message here is that, first of all, the Spermatogonia, right, this for plural, the Spermatogonia have to replace themselves, okay, and they do that via mitosis. Then meiosis is going to occur in the type B cell of this mitotic division, and it's going to produce 4 genetically unique gametes. Okay, so 4 genetically unique haploid cells. And I say genetically unique because each one has a different combination of genes, of alleles from grandma and grandpa. Okay. If you guys want me to repeat anything or have any questions, by all means, throw it out there. Okay. All right, and so this is a term you might see as you're studying this. It's called genetic recombination. Again, we mix the genes from grandma and grandpa, right, that were ultimately given to papa at the process of fertilization, and this produces unique gametes. And what we're going to call them, because we're talking about making sperm, are spermatozoa. Okay, so fancy name for sperm. Note the zoa at the end. This indicates like zoo or animal-like, and essentially the zoa component of this word is just telling us that these cells can move, right? They have animal-like qualities. As we'll see, sperm are literally sniffing out the eggs, which is kind of wild, but that's what they do. They, of course, have the little flagella, so they're moving all around, kind of competing with each other, so very animal-like. All right, so let's start to connect this back to what Professor Fink told you guys in your last lesson. This is an image of essentially a cross-section through a seminiferous tubule. Okay, so remember that a testis is made up of all these squiggled tubules. They are hollow. In these tubules are where sperm are made. And so let us add this meiosis concept to the seminiferous tubules. Okay, so what we can see here is the edge of the tubule, right? So here is a section through the tubule. We are essentially looking at this little like pie wedge, okay? At the very outside of this tubule, this is where the Spermatogonia live. Okay, so remember that they are diploid, so 2 copies of the DNA, one came from grandma, one came from grandpa. Okay, we know that the process of mitosis is going to produce 2 identical diploid copies of the Spermatogonia. Okay, one is going to remain a stem cell and it's going to still hang out on the edge of the seminiferous tubule. The other one is going to proceed through meiosis. Okay, and in the process of this independent assortment, this crossing over, this cell division, all of these different things within meiosis, the cells themselves are going to physically move, right? I'm not talking about their swimming or anything. That's not happening yet. There's no reason for a sperm to swim in a man's body, right? So we're not talking about swimming yet. But these cells are actually pushed deeper and deeper and deeper into the tubule. Okay, so from the edge of the tubule closer to the lumen, right? By the time these cells get to the lumen, they should be independent gametes, and then they can kind of be washed away into the epididymis. Okay, so here we see the type B daughter cell. This is also diploid. Once it moves into the next region of the seminiferous tubule, we can call it the primary spermatocyte. Okay, so primary indicating that there's going to be a secondary here soon. Spermato, right? So something to do with sperm site cell. So it's not an active sperm yet. It's just a precursor to the sperm. Okay, the process of meiosis 1 is completed, ultimately resulting in 2 secondary spermatocytes. Okay, so these cells have partially divided, right? So they have pulled 1 copy of DNA into each of these cells, right? But as we can see here, the cells themselves have not actually been separated just yet. Okay, so they don't complete a 100% cytokinesis. They're still kind of like gelled together. Okay, each secondary spermatocyte is then going to undergo meiosis 2. Right, so we see 2 cells, each one divides into another cell or another 2 cells, producing ultimately 4. Right, again, these cells have not completely divided yet. Each one contains its own copy of DNA, just 1 copy of DNA. But the cells themselves are not yet separated, and surely these are not yet sperm. Okay, so these cells are called spermatids. Okay, so again, not quite sperm, but genetically they are sperm. Okay, so they are non-motile. They do not yet have flagella. Okay. Finally, these spermatids, right? They've completed meiosis. They are genetically sperm, but not functionally sperm. These are then going to complete the process of spermiogenesis. Right, so it's kind of a subtype of spermatogenesis. The whole process is making functional sperm. The first part is meiosis. The second part is making them functional, making them able to swim, kind of cutting them apart so that they can swim independently. Okay, so spermiogenesis is going to begin with kind of the slicing and trimming of this excess cytoplasm. Right, and that's actually done by cells of the seminiferous tubule. Right, we'll get to that in a moment. And ultimately adding a couple kind of extra features here, right? So as we'll see on, I believe the next slide, there are 3 different parts of an individual sperm, and all of those have to be kind of activated and kind of charged up, as we'll see. Okay, so again, this is the beginning of spermatogenesis. It starts with a spermatogonium completing mitosis. The type B daughter cell then completes meiosis 1 and 2, leaving us with spermatids. Right? The spermatids are ultimately kind of trimmed and liberated, and as we can see all the way at the bottom here, this line here is showing us the edge of the lumen, and so these cells are then washed up to the epididymis. Okay, and the epididymis is where the remainder of spermiogenesis is going to occur, and storage of these ready-to-go sperm. Okay. Ultimately, when the sperm are mature, we will call them spermatozoans, right? So zoa meaning they can swim, they can smell, they can do all sorts of different things. Okay. All right, and so as I said, we are going to talk about the cells kind of surrounding this process, right? So these cells are kind of embedded within the seminiferous tubule, and so the cells that are kind of helping this process along are called two things, of course. They're called Cystenocytes because they're literally sustaining cells. They're sustaining this process of spermatogenesis. And the more antiquated term is Sertoli cells. You will see this written both ways. Okay, so let's talk a little bit more about these Cystenocytes. So on this slide, we see just the process of spermatogenesis, and all of this white is just kind of left to the imagination. Now, let us look at what is actually in that white space. For some perspective, here is the edge of the seminiferous tubule. So essentially looking at this little chunk through the tubule right here. Remember that the entire testis is made of squiggled little seminiferous tubules, right? Sperm, of course, are made, you know, kind of at the edge, and they're pushed farther and farther into the tubule. And once they get into the lumen, they are then washed up via peristalsis, right? There's not swimming yet. And ultimately, these seminiferous tubules will drain into the epididymis. Okay, so that's kind of where we are talking about here. The dividing cells, right? So the primary and secondary spermatocytes, the early and late spermatids, all of these are embedded within these yellowish cells that we see in this diagram. Those cells are called Cystenocytes or Sertoli cells. Also, they're sometimes called nurse cells, right? Why name something once if we can't name them 2 or 3 times? These Cystenocytes have tons of different roles, and they're critically important in not only spermatogenesis, but a lot of other things as well. First of all, they're important in actually pushing the developing sperm down into the lumen, right? So as I said, these cells are physically moved closer to the lumen, and the cells responsible for doing that are the Sertoli cells. Also, these nurse cells regulate how much spermatogenesis is occurring at all. Okay, so as we're going to see in a couple slides here, cystenocytes are responsible for producing a chemical that is going to increase spermatogenesis, right? So this process is all regulated by the Hpg axis, hypothalamus pituitary gonadal axis, but what is actually receiving that message from the brain? Well, the Sertoli cells. Right, so a hormone is going to bind and in response produce a chemical that increases the amount of sperm creation. Okay, specifically, the hormone that is important to the cystenocytes, and we'll look at this again, is FSH. Okay, so we're going to be talking about FSH and LH. This hormone is S, right, FSH, which binds to the cystenocytes S, also called Sertoli cells S, right? We're going to see LH, right? And that's going to bind to L things. Okay, so FSH binds to the cystenocytes. In response, the chemical it produces is androgen binding protein. Androgens are things like testosterone, okay, and a bunch of other things as well. But in this case, FSH triggers the Sertoli cells to bind or to produce ABP. ABP binds to testosterone and keeps lots and lots of it in the testis as opposed to circulating elsewhere through the body. And that testosterone, which is held in really high concentrations within the testis, is going to directly stimulate spermatogenesis. Okay. As we're also going to see hopefully today, testosterone and estrogen are literally one reaction apart. And it's not just testosterone that is going to regulate sperm production or even secondary sex characteristics, but also estrogen. Okay, so testosterone and estrogen are present in both males and females. We generally think of testosterone as being the male hormone and estrogen as being the female hormone, but both are present in both males and females to varying degrees. And there really is no black and white, right? There's always some kind of a gray area here, right? So both are made in both types of people. Okay. Sustenocytes are also really important in creating an impermeable barrier. Okay, and so that's what these are showing right here. There are tight junctions between the Sertoli cells that literally form two different compartments. Right, these compartments are essentially the outside one, right, the basal compartment, the one that's on the edge of the seminiferous tubule, and the one that's inside closer to the lumen, the adluminal compartment. Okay. And so this is really important because all of the cells that are in the basal compartment are diploid cells. They are genetically identical to any other cell within the man's body. And so their protein fingerprint, right, their MHCs on the outside of these cells are exactly what all of the rest of them within the Papa's body here look like. And so the immune system learns to recognize those cells as being just like any other cell within the man's body. However, as soon as these diploid cells cross over into the adluminal compartment, they are genetically different, right? They purposely are genetically different so that the sperm are ultimately unique, right? So these tight junctions prevent any immune system cells from essentially looking at these genetically unique cells, right? They no longer look like Papa cells because they have mixed up their genome, and keeping these immune cells out so that they don't attack the developing sperm. Right, so this is really important because in a man or in a male, his immune system develops, right, learns self versus non-self within the first couple months of life, just like anybody else. But at that time, he is not completing spermatogenesis, right? So those immune system cells are never taught that, hey, these sperm over here, they're okay, right? Don't attack them. Okay, spermatogenesis doesn't actually begin until puberty. And at that point, the immune system cells can recognize self cells as opposed to these developing non-self cells. Okay, it's a little bit different in a female because she actually starts meiosis before puberty. She starts it in utero, actually. Okay, so this blood-testis barrier is really important in preventing the destruction by your own immune system of developing sperm. Okay, Sustenocytes are also really important in literally nursing these developing cells. So they are going to provide nutrients and signals to the developing sperm. And so this image is kind of upside down from what we've been looking at. Here's the edge of the seminiferous tubule. Here are the developing, the type A and B daughter cells after mitosis. Okay, the Sustenocytes then are responsible for adding a series of proteins within the membranes of these developing spermatocytes. Okay, and so these junctions are, first of all, what help the movement, right, closer to the lumen, but also they can be channels as well. And so the nurse cells can actually take sugar and such from inside them, right, which they have, of course, converted into ATP and whatnot, and they can literally feed the developing spermatocytes. Okay. Also different types of signals, right? So ABP, for example, is triggering the further division of these different cells. Okay. The Sertoli cells are also responsible for phagocytosis, right? So actually like munching of the extra stuff, the extra cytoplasm of these cells. Okay. They can provide apoptosis signals, right? So essentially they can assess whether this process is proceeding the way it should, right? So if cell division happens in a faulty way, and so one sperm ultimately has too many chromatids or not, excuse me, or not enough chromatids, they can be triggered to eliminate themselves. Okay, also, excuse me, if there is too much estrogen, there's a lot of different reasons why that would be, and hopefully we'll get into that today. But if there is too high of estrogen, apoptosis signals will be provided as well, right? So if there is an excess amount of the female hormone, spermatogenesis is going to slow down based on the feedback from the Sertoli cells. Okay. And finally, the Sustenocytes are going to secrete testicular fluid into the lumen for the transport of sperm. This is not semen. Okay, this is the fluid in which these little non-motile or immobile cells are going to be suspended so that they can actually be pushed along through the seminiferous tubules and up into the epididymis. Okay, so this is really just to sustain and transport the sperm. All right, so let's look at some histology of the seminiferous tubules for a moment. Just for a little bit of perspective, we can see the circle part here. That is a seminiferous tubule, which is right next to another seminiferous tubule, which is right next to another seminiferous tubule. So really the entire testis is made up of densely packed tubules that make components of the semen, right? Specifically the sperm itself. Okay. There are spermatogenic cells within the tubule itself. So that's what we just looked through. Remember that at the surface in the basal compartment, that's where the diploid cells are, the Spermatogonia, and ultimately they divide as they get closer to the lumen, right? So we can even see in this histology the different chromatids and where they are in their different phases of development. Okay, embedded, or these spermatogenic cells are embedded within the Cystenocyte. So that's the lighter pink here. Okay, so those are the Cystenocytes or Sertoli cells. Around each one of these seminiferous tubules are cells called myoid cells. All right, remember the root MY, MYO, MYS, this always means muscle. And so there is a very thin layer of smooth muscle, okay, around each one of these Sertoli cells, which is responsible for producing peristalsis of the testicular fluid and the spermatids within them. Okay. Also, in between here, in between the round seminiferous tubules are other types of cells. These are called interstitial endocrine cells, right? So interstitial, it is not actually inside the seminiferous tubule, it's in the outside, in the interstitial fluid. Endocrine, right? They have something to do with making hormones, right? Which you may remember from Professor Fink's talk. And these guys are also called Lydic cells, right? So the L is going to be important in here because it is responsive to the hormone LH, luteinizing hormone. Okay, these cells are responsible for producing the sex steroids, including testosterone, but not only testosterone, in response to LH. Okay, so LH binds to Lydic cells, FSH binds to Cystenocytes or Sertoli cells. Okay, which leads us to talk about the Hpg axis. Okay, so way back in this semester, we talked about two hypothalamic anterior pituitary gland axes. Okay, we talked about the one that regulates the thyroid gland. We talked about the one that regulates the adrenal gland. Today, we get to talk about the final hypothalamic anterior pituitary axis and how it regulates spermatogenesis. Okay, so again, Professor Fink talked a teeny bit about this, but I want to elaborate on this and connect meiosis with the production of hormones with all of the other things that are happening here. Okay, so the Hpg axis includes the hypothalamus. That's what kind of starts this whole process. It releases gonadotropin releasing hormone, or GnRH. And this really is a lot of alphabet soup. Welcome to the endocrine system, right? Perhaps you remember from earlier this semester that tropic hormones cause the release of hormones from another organ. In this case, this hypothalamic hormone is ultimately going to lead to the release of hormones from the gonads. But before it does, it needs to release other hormones. Okay, so remember that all these releasing hormones are coming from the hypothalamus. The hypothalamus releases GnRH into the first capillary bed of the portal system. It is taken down to the anterior pituitary gland. Remember, that's kind of right above the roof of your mouth. Okay, and in response, the anterior pituitary is going to release the gonadotropin hormones. So the hormones that are going to lead to the release of other hormones from the gonads. Like, there are two gonadotropin hormones. LH is luteinizing hormone, and FSH is follicle stimulating hormone. Both of these hormones were named for the actions that they induce in females, but both hormones are exactly identical in males as well. Okay, so again, the luteinizing and follicle stimulating, those terms are referring to the female actions, but they're also active in males. Okay. When these two hormones are released into the blood, they are going to be, of course, distributed throughout the body in the circulatory system, and they're only going to bind to cells or organs with receptors for LH or FSH. In this case, those receptors for LH and FSH are only present within the gonads. All right, so the gonads are the testes in males and the ovaries in females. Note that testis, this is singular, testes is plural, and this is the gonad itself, not the scrotum, not the blood vessels or lymphatic, nothing like that, just the testis itself. Okay, so LH, as I've mentioned, binds to the Lydic cells, and in response, the Lydic cells are going to produce testosterone. Okay. The Sertoli cells, right, or these Cystenocyte cells, are going to produce androgen binding protein, okay, which we talked about a little bit before. Again, LH binds to Lydic cells, FSH binds to Sertoli or Cystenocyte cells. Okay. In addition to releasing androgen binding protein, the Sertoli cells are also going to release a hormone, right? So gonadotropin hormones, they're going to release lots of hormones. So inhibin is yet another hormone that is going to be released in this process. As the name suggests, this is going to inhibit something. It's going to put the brakes on something. Okay. So just like any of the other hypothalamic anterior pituitary axes, this axis is going to be regulated by negative feedback, right? So the more testosterone and inhibin are present in the blood, the more we're going to have kind of the brakes be put on the hypothalamus and anterior pituitary gland. Okay, so here is a very basic summary. I know you might not believe it right now, but very basic summary, right? Hypothalamus releases GnRH. GnRH tells the anterior pituitary to release these two gonadotropin hormones. LH binds to the Lydic cells, producing lots of testosterone. The more testosterone is released, the more this hormone is going to suppress further secretion of the hypothalamus and anterior pituitary. Okay, so testosterone itself is going to be regulated so it doesn't get super high by exerting negative feedback farther up the axis. Okay, FSH binds to the Sertoli cells, right? And the Sertoli cells, of course, release ABP. We'll get to that again, but they also release inhibin. Inhibin can put the brakes on the anterior pituitary gland, right? Further suppress the release of more gonadotropins. Okay, LH and FSH themselves can also exert negative feedback farther upstream. Okay, so lots of negative feedback. The body really cares about not having excessive testosterone present. And so if you take testosterone, right, so if you are taking anabolic steroids, right, so that your muscles get nice and big because testosterone promotes muscle growth, essentially that excess testosterone is going to inhibit your own hypothalamus and your own anterior pituitary gland. Okay, so yes, you will have big muscles. Yes, you will have super high testosterone levels, but you're not going to have LH and FSH be released anymore. Okay, and so remember that FSH is responsible for telling the Sertoli cells to release ABP. ABP directly stimulates sperm production. Okay, and so a lot of times what you see is individuals who are taking steroids have very low sperm count, even though they have so much testosterone circulating in their system, because negative feedback inhibits the release of FSH. FSH is needed to make sperm. Okay, so that is what, that's why steroids can inhibit reproduction, right? Even though it is a sex steroid. Okay. One final note before we move on to the next cell, these Lydic cells release testosterone. Testosterone itself has its own effect on other target cells throughout the body. So things like secondary sex characteristics, so the development of a beard, the development of muscles, of course, right? Larger muscles on average than female muscles. The development of kind of a broader chin, right? Behavior, libido, all of these different things are affected by testosterone because cells throughout the entire body have testosterone receptors in both males and females again. All right, so let's kind of pull all of these different pieces together. How do all of these crazy hormones regulate spermatogenesis? All right, so mitosis, then meiosis, and then spermiogenesis. What's happening? Okay, here we see the hypothalamus way at the top. Here's the anterior pituitary gland. And of course, these arrows are essentially kind of fast forwarding through the entire circulatory system to a cross section through the seminiferous tubule in the testis. Okay, so that's what's going on here. Surely the brain and the testis aren't physically close together, but they are talking back and forth all the time in so many ways, right? Okay, so GnRH released into the portal system, which immediately is delivered to the anterior pituitary gland. The anterior pituitary, in response, releases both FSH and LH. Right, so we can see them being released into the blood here. FSH only binds to cells with FSH receptors, specifically the Sustenocyte or Sertoli cells. In response, the Sustenocytes are going to release ABP into the interstitial fluid, into the surrounding area. ABP is going to bind to testosterone. Where does testosterone come from? LH stimulates the interstitial endocrine cells, which we also know are called Lydic cells, stimulates them to release testosterone. Testosterone binds to ABP. And because there is so much testosterone in the seminiferous tubule, right, it's held there as opposed to just circulating throughout the body, this is going to stimulate spermatogenesis. Okay, I want to elaborate a little bit more on how testosterone is made by the Lydic cells. And to do so, I want to point out this metabolic pathway. Note that all of these steroid hormones are derived ultimately from cholesterol, right? So cholesterol gets a super bad reputation, but we need it for so many different things, right? So you need it to regulate your blood pressure, you need it to make your cell membranes, you need it to make sex steroids, right? So cholesterol is super important, just not too much, right? So cholesterol can be modified by a series of enzymes into all of these different chemical structures. Note that each of these red arrows is indicating a chemical reaction. So cholesterol is modified by an enzyme to produce pregnenolone. All right, pregnenolone can then be modified by either an enzyme called CYP17 or an enzyme called 3-beta-HSD. These are not test worthy, by the way. But what I'm getting at here is that this hormone can have two different fates, right? Or actually a lot more than two different fates, depending on whether it is modified by CYP17 or 3-beta-HSD. Okay, and so this is how our bodies can regulate how much of any of these different hormones we have. We can turn on the CYP17 gene, or we can turn on the 3-beta-HSD gene. Okay, and so all of these genes are tightly regulated by hormones and by different types of factors, including genetic factors within our body. Okay, and so again, we can kind of shift this metabolic pathway in a lot of different directions based on the presumed need of the body at the time. Okay. So again, transcription of different genes leads to different synthesis pathways, right? So we can end up with aldosterone or cortisol or progesterone, estrone, which is a type of estrogen. Estradiol is the more powerful type of estrogen, and testosterone. Androstenedione is another one, another androgen that's super potent, super important. Okay, but what I'm getting at here is that the turning on of different genes is regulated by hormones, right, such as LH and FSH binding to a receptor and therefore leading to the transcription of each one of these genes. Okay, note that LH, when it binds to receptors on the Lydic cells, it is going to initiate a pathway within these cells of the testis to ultimately turn on the gene that is responsible for converting androstenedione into testosterone. Okay, and so that is how you get testosterone, that one single enzyme right there. Okay. On a similar note, something that I'm going to loop around to again later in this talk is that one particular gene, the CYP19 gene, produces an enzyme called aromatase, right? So CYP19 is the DNA code. Aromatase is the enzyme that is actually doing the folding, cutting, etc. When CYP19 is turned on, right, so it is expressed, transcription, translation, etc. CYP19 is going to convert any existing testosterone available into estradiol. Okay, so estradiol is the potent estrogen. And so even if you are a man, your testosterone can be converted into estrogen by turning on one single enzyme. And this can happen with hormones that are endogenous in your body, right? So hormones that you yourself are producing, they are going to make a little bit of estrogen to balance out the testosterone. That's a natural thing. But also there are tons of chemicals that we interact with every day. Some of us more than others, some chemicals more than others. And those chemicals, in ways that we're going to look at very soon here, are going to increase or decrease the amount of this gene, right? Or how much this gene is turned on or turned off. And so you might have heard of endocrine disruption before. Essentially, this gene can be upregulated, right? It can be turned on to produce tons and tons and tons of aromatase, which takes all existing testosterone and it's going to convert it into estrogen. Okay, so A couple take-home messages here. First of all steroid hormones derived from cholesterol, whether we're talking about stress hormones, whether we're talking about hormones that regulate salt balance and blood pressure, or we're talking about sex steroids down here. And this pathway can be shifted in lots of different directions. For example, if you are super stressed out, more cholesterol is going to be diverted to making cortisol than it is to making these over here. And so oftentimes a prolonged stress response can lead to hormone imbalances with particularly estrogen throughout your body, again, because more cholesterol is used to make stress hormone than it is to make estrogen. Okay. Lots of soapboxes today. Okay, so one enzyme determines if you have testosterone or you have estrogen. Estrogen is also used in the male reproductive system, in the male body, at very low levels, of course. And some of the ways that it does influence the male reproductive system. First of all, penile vasculature, right? So the blood vessels that deliver blood into the penis itself and lead to an erection are lined with estrogen receptors. So they are susceptible to behavioral changes based on the presence of estrogen. And so if you have too much estrogen in your body, based on endocrine disruption or some other kind of issue, this can inhibit the ability of blood to engorge the penis and therefore produce an erection. Also, Lydic cells, Sertoli cells, and the germ cells, so the spermatogenic cells themselves, they all make estrogen at some point during the developmental process. The problem comes in where there's too much estrogen, right? You want a very low level of estrogen in order to promote these processes appropriately, but not too much because that will lead to more feminine traits. Okay. And finally, there are regions of your brain, actually maybe more than you'd think, that have estrogen receptors as well. And so estrogen levels varying over the course of a menstrual cycle, for example, or just due to exposure to different chemicals or whatever, this can very much affect your behavior. Specifically behaviors regarding libido, but more than that as well. So again, it's not that the male hormone is testosterone and the female hormone is estrogen. We both need both hormones. It's just males have more testosterone and females have more estrogen. Coming back to the Hpg axis and how it regulates spermatogenesis, right? We know that testosterone, which is from these interstitial cells out here, is going to produce more testosterone in response to LH binding to them. Okay. As I've said, this extra testosterone in the area, right, in the testis itself, is going to lead directly to spermatogenesis. The way that testosterone is increased within the seminiferous tubules is because ABP is going to bind to it and, of course, increase that concentration in the area. So when you are thinking about which hormone stimulates which process, FSH is the hormone that directly increases spermatogenesis. LH directly stimulates testosterone. Okay. Testosterone itself, of course, has a lot of different effects throughout the body, including on behavior. It influences the maturation of the sex organs themselves. Right, so changes during puberty, changes during fetal development even. Having just the right levels of testosterone versus estrogen is critical in developing a penis versus a clitoris. Right, we're going to look at that here shortly. Okay. Testosterone is really important in the development and maintenance of secondary sex characteristics. As I was saying before, the development of a beard, of a wider jaw, of thicker muscles, right? All of these things are secondary sex characteristics, which means that they are not directly involved in reproduction, that is sperm meeting egg, but they are associated with sex steroids and in fact produced by sex steroids. And also they are symbols to the opposite sex or to the same sex that this individual is masculine. This individual is producing sperm. Okay, so it is very easy to look at male versus female and recognize these characteristics, right? Breasts versus beard indicates male versus female. Again, the levels of these hormones, or these hormones don't necessarily produce strict male, strict female. There is actually a lot of gray area in between. Actually less so in humans than there is in most other species. Usually the identification of, male versus female or even something in between is a lot more fluid in other types of animals and to an extent in humans as well. Right? It's not all just black and white. Okay. As I mentioned before, testosterone is going to determine erectile function, right? So too much estrogen over testosterone is going to lead to less blood flow into those, or less vasocongestion and therefore erectile dysfunction. Okay. Testosterone is generally associated with more aggressive behavior. Okay. But again, there's a wide range of effects and libido itself. Okay. Of course, too much testosterone is not good either. And so this entire axis is regulated by negative feedback, just like all of the other axes that we've talked about. We see that a lot of testosterone is going to inhibit the anterior pituitary and also to an extent the hypothalamus itself. Inhibin from the Sertoli cells or the Sustenacites is also going to exert direct negative feedback. Okay. Oops, sorry about that. So in summary, GnRH leads the anterior pituitary to secrete LH. LH stimulates testosterone. FSH triggers the Sertoli cells to release ADP, which binds to the testosterone made by the Lydic cells to stimulate spermatogenesis. We know that the Spermatogonium is going to undergo mitosis, producing two identical daughter cells. One remains in the basal compartment, right, to make more cells ultimately. The other is going to proceed down into the adluminal compartment, right, which is isolated from the immune system so that these cells can become genetically different without being attacked by the immune system. The type B daughter cell becomes a primary spermatocyte. It completes meiosis 1, producing two genetically unique secondary spermatocytes, which themselves enter into meiosis 2. Each one of these daughter cells is going to produce two, again, genetically unique daughter cells itself. Ultimately, one primary spermatocyte produces 4 of these early spermatids. They are genetically sperm, but they are not functionally sperm yet. These early spermatids then begin spermiogenesis, which is essentially making them into functional sperm. Okay, the Sertoli cells ultimately trim these functional sperm, right, making them separate, making them kind of streamlined, and then they release these spermatozoa, right, animal-like sperm, into the lumen, where they are then washed away via peristalsis into the epididymis. Okay. This process takes, give or take, 2 months. Right, so about 64 days is how long it takes to go from a single stem cell all the way down to functional or ready to go, ready to swim sperm. Okay, let's look at some of the things that have to happen to one of these little spermatids before it can be called a functional spermatozoan. Okay, the round spermatid in the seminiferous tubule is ultimately going to elongate and mature to an extent. Okay, so here we can see it's starting to elongate, and so it's not a round little clunky thing. Ultimately, the sperm itself is going to have 3 distinct regions. Okay, first of all, there is a head. Okay, the head is on human sperm, cone-shaped. And I say on human sperm because each species has a different shape of sperm, which helps to prevent interspecies fertilization. Okay, so there are very few instances where sperm and egg can actually be fertilized in different species, right? So this is very unique to humans. The head is triangular in shape and it has this like little helmet on it. The helmet is called an acrosome. And essentially, as we're going to see in a future lesson, this acrosome is really important in essentially like drilling through the different protective membranes of the human egg. Okay, this acrosome is filled with a lot of different enzymes. So as the sperm is kind of like wiggling through those different membranes, the acrosome is spitting out digestive enzymes, which kind of makes a little tunnel so that the sperm itself can wiggle through to actually access the egg and penetrate its surface. So the acrosome, the acrosomal reaction, which we'll talk about in a future lesson, is critical in allowing fertilization. Okay, the mid piece, right? So this here is the mid piece itself. It's pretty much a bunch of mitochondria all kind of packed together. And the reason for this is that the life of a sperm is up to 5 days after ejaculation. And so there's no time for the sperm to look for food, right? So there are some nutrients in semen itself that can nourish the sperm, but for the most part, the sperm is not going to stop and order breakfast. It has to be equipped with as much energy as it needs to swim with all its might for up to 5 days. Okay, so the mitochondria are there to essentially, take this lunch that was packed for it and convert it into ATP. That ATP is then used to make the flagellum beat back and forth. And of course, it is doing this constantly for up to 5 days at a time. So there really has to be a lot of ready energy in the sperm so that it can make it to the egg. Okay, without eyes or anything, like it's just, not asking for directions or anything, it's just going. Okay, and of course the tail portion, which you think of when you think of a sperm, this is the flagellum, and essentially there are little mechanisms, little proteins within this kind of joint here that allow the tail to swing like this. And so it is just kind of swimming around more or less blindly, although, as I said, it can sniff out the egg. To be continued. All right, so some healthy parameters. We can see that the testosterone levels in males is naturally going to decline over time. Right? In general, all of your cells get a little bit tired over time, and so after they've been cranking out testosterone, for decades, they start to slow down a little bit. Okay. All right, so some seminal content, so contents of semen, the volume is generally 1.5 milliliters or more. The pH is a little bit alkaline because sperm really like an alkaline environment. Keep that in mind because pretty much the entire female reproductive system is very acidic. And so the semen has to actually change the pH of her internal environment in order to promote longevity in the sperm. Okay. In every milliliter of semen, there are at least 15 million sperm. Okay. So that is healthy level. Lower than that is considered infertile. Right. Not that there aren't sperm necessarily. It's just that 15 million is per milliliter, right, is what it takes to actually increase the chances of sperm finding egg. Okay, total motility, so about 40% or more of those 15 million sperm have to actually be able to wag their flagella, right? Healthy levels are almost 60% alive. Okay? For the most part, sperm are stored within the epididymis for weeks, maybe months at a time, and so they do die off in that time, and so a healthy level of live sperm is considered to be about 50%. And white blood cells less than 1 million per milliliter. Okay, so just a couple healthy parameters of sperm and semen, and which going to elaborate just a little bit more on. I've alluded to this a couple times, but I want to talk a teeny bit more about endocrine disruption. There are many chemicals that we interact with on a daily basis that can affect these tightly controlled axes. Okay, so normal response, right? We have, hormone LH, FSH, whatever that is released into the blood. It binds to a receptor and ultimately produces some kind of cellular response, right? We make testosterone or sperm or whatever in response to normal levels of this normal old hormone. Some of the chemicals that we interact with can mimic hormones, right? So even when our own bodies are releasing just a little bit of LH or FSH, right? And this is applicable to males and females. These chemicals can bind to the same receptors and induce the same response that our normal hormone does. So maybe our body only wants this much of a response, but these extra mimics are producing a much stronger response than our body would need at that time. Also, some of these chemicals can literally block hormones. Okay, so instead of binding to these receptors, right, and therefore producing a response, instead these chemicals can kind of plug up the receptors. So even if we do release normal hormones, right, they are not going to be able to produce the same response, right? So no matter how much FSH or LH, you're not going to get any testosterone produced because these receptors have been blocked. And so when I talk about chemicals, the list is horrifying, but one of the many reasons why organic food is so popular is that the residual pesticides that are used to, of course, make agriculture more productive and therefore feed more people. These pesticides can linger on your food and can act as hormone mimics or hormone blockers, right? And a lot of them do this. Flame retardants. So that new carpet smell? Well, that new carpet smell is literally a chemical that is sprayed on your carpet to reduce the chances of it catching on fire. Same thing with mattresses, same thing with a lot of clothing products, even all kinds of different furnitures. These substances are very flammable. And have caused a lot of deaths over the years, especially if if individuals are like smoking in bed or something. So these chemicals are there to protect us from fire. Of course, a wonderful thing. There's a huge demand for this, but breathing in these chemicals, touching these chemicals and having them seep through your skin, which they're doing all the time, can plug up receptors in your thyroid gland, for example. And so lots of endocrine disruption linked back to these chemicals. Plasticizers, so chemicals that make your water bottles nice and squishy without breaking. And a lot more things than that are also disruptive to your endocrine system. Pharmaceuticals can purposely affect your endocrine system, right? So a lot of pharmaceuticals, as we've talked about over this semester, can literally mimic hormones, can literally on purpose block receptors. So they do this on purpose, but there are many that are having a lot of side effects, some unintentional side effects. And so actually, whenever you listen to drug commercials, there's always the, oh, they do these things, hooray, it's a miracle drug. But then here are these 17 other side effects that this drug is responsible for producing. Well, usually what that is, or what these things are caused by, is that, yes, the pharmaceutical is going to, block up the beta adrenergic receptors, for example. Okay, that's great. That's going to decrease your blood pressure. But where else do we have those receptors, right? So when this drug binds to those other receptors, it's going to have unintended side effects on those other tissues, right? Just because wherever there are receptors for these different things, those are the tissues that are going to be affected. Okay, and finally, emulsifiers. These are chemicals that are in, for example, hand soaps. And so these are great at emulsification, right? So breaking down lipid-rich substances. They help you to wash your dishes. Hooray, this is great. But a lot of them are also endocrine disruptors. And so a few years ago, when the huge BP oil spill was happening and everyone was going down to Louisiana to take Dawn dish soap and like scrub off the ducks and everything. Like, yes, the oil was removed from the poor little ducks. Hooray. But at the same time, we were dumping so many of these endocrine disrupting compounds into the water. So yeah, helping on one hand, but there's always unintended side effects. Yay. So a lot of these different chemicals can cross the placenta, they can cross into the breast milk. And so we are essentially building up these compounds within our bodies, most of which are lipid soluble, right? So for example, this here is what estrogen looks like. This is one of the chemicals I'm talking about called BPA. Keep that in mind. And so this mimics estrogen and does a couple of other things as well. All of these things are soluble in lipids, so they stick around in your body for a really long time, accumulating higher and higher and higher levels. And if you go to have a baby, your placenta, right, the cells within your mammary glands do not recognize that this isn't this. Right. And so these chemicals, which are brand new as far as our body is concerned, and they really are new within the past few decades, they are allowed to go to baby. And of course, baby is very small, right? So a smaller concentration of these chemicals can have a much bigger effect. And these things can mimic hormones, and hormones expressed at different levels at different points in development has a huge impact on whether, for example, you become a boy or a girl. Right? So very big implications here. These things are especially disruptive during development. I want to show you this little graph here. This is gestation, so essentially weeks of a pregnancy. We can see that in about week number 9, the testosterone levels in the fetus, so this is a boy, are going to increase dramatically, right? So if, mom interacts with lots of BPA and everything in, this period of pregnancy, maybe it doesn't have a huge effect. But if she, if her body is kind of dumping BPA and all these other chemicals into, or across the placenta into her developing boy, that is going to throw off the balance between the testosterone levels produced by her baby boy and the estrogen that is going to be surrounding it and within its blood. And we can see that as testosterone increases, right, as baby boy develops, essentially, this testosterone is going to stimulate essentially the regression of the girl parts and the development of the boy parts, right? So we all start off as females. So in order to get a male as opposed to a female, we have to get rid of the female structures. Testosterone is needed. Develop the male structures. Testosterone is needed. Testes need to descend from around the kidneys to within the groin. Testosterone is necessary. Genital tubercle formation, so the development of a penis over a clitoris. All of these things, right? External genital differentiation and growth, these all are dependent upon lots more testosterone than estrogen. Right? So really important to have just the right levels of these hormones during this part of development, right? So whatever you make during this period, that's what you're going to have for the rest of your life. Okay. And I am out of time. Guess I should get off my soapbox for today.